My name is Morgan Martin. I'm a sophomore bio major, Spanish minor from Atlanta, Georgia.
Hi, my name is Mariah Costin. I am a third-year bio pre-med major from Chicago, Illinois.
The following is brought to you by
Hey guys, maybe there's a few things in life that we need to learn. Maybe why my favorite color is blue, or things more important like why DNA is synthesized. Keep watching and you'll hear more about it.
Now, when scientists hear the word DNA, two names often come to mind: Watson and Crick. These two men were responsible for identifying the double helical nature of DNA, as well as its code being a triplet, if we were to get into codons, amino acids, and all the other good stuff. But that's not the focus for today. Today, we want to discuss two other scientists, Matthew Meselson and Franklin Stahl, and they were responsible for identifying the nature of the product of DNA replication, and that is semi-conservative replication. Like any other area of scientific research, theories were thrown left and right regarding the nature of the product of DNA replication. The three types under major consideration were conservative, semi-conservative, and dispersive replication. Conservative replication produces two double helices, in which one helix is composed entirely of the original chromosome, and the other helix is composed of entirely new strands. Conservative replication does not alter the chromosomes. However, semi-conservative and dispersive replication do. Semi-conservative replication produces two double helices, in which both contain one strand from the parent and one strand from the daughter. Dispersive replication also produces two double helices, except segments from the parent and daughter are intertwined into both strands.
That's enough walking. Now, how did Meselson and Stahl know that semi-conservative replication was taking place and not conservative or dispersive? Glad you asked. Let's take a look.
First, they had to choose an organism to study. They decided to work with a bacterium commonly known as E. coli. Meselson and Stahl realized that if they could tag parental and daughter strands of DNA in a way that would make them distinguishable from each other, they could determine which of the three types of replication were taking place. Like all organisms, bacterial cells copy their genome before every cell division. To distinguish parental strands of DNA from daughter strands, Meselson and Stahl devised a plan to grow E. coli cells for successive generations in the presence of different isotopes of nitrogen. Nitrogen 15, indicated by the red medium, and nitrogen 14, indicated by the orange medium.
If you've taken general chemistry, then you know that isotopes are versions of the same element, simply with different masses. This difference in mass, though, was the key to the experiment because it created a difference in density between nitrogen 14 and nitrogen 15 containing DNA. A technique with a very sophisticated name, density gradient centrifugation, was used to separate molecules based on their density. And so DNA containing nitrogen 14 and DNA containing nitrogen 15 would separate into distinct bands,
as indicated by the chart at the bottom of this lovely diagram, which will be explained a little bit better looking at the details of the actual experiment.
For the experimental setup, Meselson and Stahl began growing E. coli cells with nutrients that contain only the nitrogen 15 isotope. They purified DNA from a sample of these cells, which would be considered generation 0, and transferred the rest of the culture to a growth medium containing only the nitrogen 14 isotope. After enough time had passed for these cells to divide once, meaning that the DNA copied one time, they removed a sample and isolated the DNA. This sample was identified as generation 1. After the remainder of the culture had divided a second time, they removed another sample and isolated its DNA, labeling this, you guessed it, generation 2. Finally, all three samples were centrifuged separately, and the locations of the distinct DNA bands were compared in each sample to determine their respective densities. The big gray box in the middle of this diagram illustrates the concepts behind this experiment using DNA's double helical structure. The conservative, semi-conservative, and dispersive models make distinct predictions about the makeup of the DNA molecules after replication occurs in the first and second generations. The black and white photograph at the bottom shows the actual experiment's results. Generation 0 sample produced a single heavy band corresponding to the density of nitrogen-15 isotope, which was expected since the E. coli cells were originally grown in a medium containing this isotope as the sole source of nitrogen. Generation 1's sample produced a single dark band indicating an intermediate density, which was a hybrid between the densities of both nitrogen isotopes. At this point, conservative replication was ruled out because Generation 1's sample would have produced two distinct bands instead of only one if the original parental molecule was completely conserved. Generation 2's sample produced two bands, one of intermediate density, like Generation 1, and one of low density, corresponding with nitrogen-14. Had dispersive replication occurred, Generation 2 would have only produced a single intermediate density band again. These two distinct bands indicated that each newly made DNA molecule consists of one old strand and one new strand. Therefore, semi-conservative replication was strongly supported. That was a lot of information. Now that we understand that DNA undergoes semi-conservative replication, let's try to describe all three types of replication using something a little more fun. Let's exemplify the types of replication using something that everyone loves, music. And I'll hand it over to Mariah. Let's get creative.
These days, I'm letting God handle all things above me. The things I can't change are the reason you love me. These days, I'm letting God handle all things above me. The things I can't change are the reason you love me.
During the song Jungle by Drake, it is revamped by someone named GABI, and basically what she does is uses his song as a template to recreate a newer version, which would be an example of conservative replication.
I was struggling to learn what life's about. On my way, money taught me Spanish, make it on delay.
In the last clip, you heard Legend by Drake, which was taken from Genuine's So Anxious original version. And basically what he did was took the song and intertwined it while also rapping over the original song, which would be an example of a semi-conservative replication. In the last clip, we see Anaconda by Nicki Minaj, who uses Sir Mix-a-Lot, who happens to be an older artist, which would show an example of a dispersive replication. As you may know, or probably not, DNA synthesis has to happen in a certain direction, 5' to 3', which starts on the leading strand. In order to make sure DNA synthesis is successful, there are a few components that are needed to make sure that everything goes well.
Excuse me.
What? What do you want?
What is DNA synthesis? How do you do it?
Glad you asked. Let's dive in.
To begin the copying process, DNA polymerase adds the correct nucleotides at the 3' end of the RNA primer with the help of a DNA sliding clamp for stability during the process. On the other end, the lagging strand, which I would refer to as the bad kid, since it takes a bit more to make sure the process is successful. On the lagging strand, it synthesizes away from the replication fork, which exposes the gaps that are essentially filled with Okazaki fragments. Thereafter, they are filled with Okazaki fragments. DNA polymerase 1 removes RNA primers and replaces ribonucleotides with the correct deoxyribonucleotides. Then, the DNA ligase reattaches the fragments to create a continuous strand,
and it's a successful replication. But before DNA replication can be considered complete, DNA polymerase III proofreads to ensure the correct bases are in place. As a whole, replication takes place in a replisome.
Wasn't that double helix exciting? Watch our next video for more.